So an outbreak of diphtheria has occurred for the third time in a decade in a small village in South Africa. Diphtheria is endemic to the area with many villages colonized with different bacterial strains. Then vaccine distribution in this area is difficult due to treacherous terrain. A team of doctors is sent to the region to conduct a health campaign. Toxic strains of C. diphtheria are isolated from symptomatic patients. Which of the following best explains the initial emergence of a pathogenic strain causing such outbreaks? Option A, presence of naked DNA in the environment. Option B, infection with a lytic phase. Option C is lysogenic conversion. Option D is suppression of lysogenic cycle. Option E is uh, conjugation between the toxigenic and non-toxigenic strains of C. diphtheria. So we proceed to the next. So you have an outbreak of diphtheria in the village. Okay, so the main thing they're asking is the toxigenic strains of diphtheria are isolated from the patients. Which of the following is responsible for the emergence of the pathogenic strain? All right. So one thing I remember is that I have talked about this in all my previous sessions, right? But for everyone who is new, we'll discuss this again. Yes, excellent. So this is basically your lysogenic conversion. Lysogenic conversion is basically a type of transduction. All right. Um Transduction, which is a specialized transduction example. And if people remember, I had given five bacterial toxins which lead to toxigenic strain formation due to lysogenic conversion or your specialized transduction. There are five bacteria in which the toxigenic strains appear because of that. Can anyone name the five to me? I remember telling it because I remember telling this mnemonic A, B, C, Ds. So the ABCDs of bacteria or something, whatever you want to remember it as. So, okay, excellent. I'm getting answers. So A stands for your group A Streptococcus. All right, right. B stands for your botulinum toxin. Okay, by Clostridium botulinum. All right. C stands for cholera toxin. Right. And D stands for, like we have talked over here, diphtheria toxin. All right. And S stands for Shiga toxin. All right. Is it clear to everyone? A, B, C, D, S. The ABCDs of bacteria or bacterial toxins, anything you want to remember. A stands for group A Streptococcus, B stands for botulinum, C stands for cholera, D stands for diphtheria, and S stands for Shiga toxin. Am I clear? This is what you have to remember. Okay. Then you have to remember one more thing related to this. So all these toxins have two parts. That is A and B. So most of these toxins, okay. So I won't generalize it. I'll say most of these toxins have A. Let's talk about diphtheria in particular. So A stands for active, B stands for binding. This is a way to remember, right? So by binding, what I mean is this part of the toxin B, binds to the cell walls and cell membranes, okay? And toxin A is the active part which enters it. In case of diphtheria, it leads to ADP ribosylation. All right. Am I clear to everyone? All right. So you have to remember this. This has been a persistent question in all my last three sessions. So that is why I have been highlighting this. One more thing, I think someone asked me last time. I have written it down. All right. So I think I had told you before that we have two kinds of transduction, which is your generalized and specialized transduction. Generalized is known as your lytic phase and specialized is your lysogenic phase. All right. So one thing you have to remember is what happens in the lytic phase. The virus infects the bacteria, but it's the viral genome. What happens is viral genome is not integrated into the host genome. All right. Instead, the viral genome replicates inside the bacteria. Okay. So what happens is I'll draw it out. So this is your bacteria. There comes a phage. Uh, Phage comes and attaches to it. A viral phage with its genetic material. Okay. what happens is this genetic material enters the bacteria. There is genetic material inside the bacteria also. Now, what happens is viral particle enters, it splits out the bacterial genetic material, and then you have this is your bacterial genetic material, and this is your viral genetic material. All right. So what happens is there is a randomized mixing of them, and it forms basically more viral particles with these genetic materials, right? So there are multiple virons inside this, and eventually what happens is the cell bursts. Once the cell bursts, these viral particles come out, and they further form phages and spread out. All right, is it clear? This is basically a lytic kind of phage, lytic transduction. All right, and lytic phase usually helps in spread of an infection, not in the formation of toxins. So when it comes to formation of toxins, I'll show it again. So you have your bacteria over here. Okay, this is bacterial genetic material. This is your phage which attaches with the genetic material. Now this is the genetic material of the phage. Now what happens is it enters the bacteria and it binds along with the bacteria. This is the phage genetic and these are the bacterial genetic material. Now what happens is this basically this is the infectious part of the bacteria which binds to the viral genetic material. So what happens is this multiplies and replicates with the infectious part of this bacteria, and then it develops into phages inside. Again, the same thing happens. These phages with the infected bacterial genetic material attached to the viral material, they come out of the cell and they go and infect the other cells. And once this bacterial genetic material enters into this cell, it gets attached over there into its incorporated into its genetic material, and this becomes a toxigenic strain of the bacteria. Is it clear?
Thanks for watching. Please give a thumbs up and subscribe for more.